Dear participants, first of all, I would like to extend my thanks to Dr. Madhu Yashpal and the Institute for giving me this opportunity. After the lecture of Professor Ratnakumar Pati, I am sure that you have understood the concepts of basic chronobiology. In my lecture, I will be speaking about the applied aspects of chronobiology, particularly cancer chronotherapy. So first of all, I will speak about chronotherapy. As you see from the slide, that chronotherapy is a subdiscipline of the chronobiology. So chrono means time and therapy means treatment. So treatment given in relation to time, or you can define the chronotherapy, that is the use of circadian or other rhythmic cycles in the application of therapy. So then how this concept or chronotherapy is evolved? So it has been seen that the onset of health problems and its severity exhibit variations along 24-hour time scale. So this onset of several acute medical conditions is correlated with the circadian phase. It means the severity is time dependent. In one time point, the severity of health problem may be maximum, in other time point it may be minimum. So then with this, the experiment or the research in the field of chronotherapy, so perhaps the experiment by Seving et al. In 1974 is the first experiment that establishes the concept of the chronotherapy. Here you can see that this, or we can say that Seving et al. They used a chemotherapeutic agent, that is the arabinocyl cytosine, and they examined the toxicity of this agent in mice, and they studied the percent mortality on account of this drug. So what they did, they divided the mice into 5 groups and delivered this arabinocyl cytosine at 5 time point of the day. So in the first group, the dose of the drug was 200 mg per kg of body weight, and it was delivered early morning, or we can say the rest span of the mice, because these are the nocturnal animal and during the daytime they are inactive. So rest span of the day the drug was delivered. In the second group, the dose was the same and it was delivered in the afternoon hours. In the third group, the drug was delivered at initial phase of the dark. In the fourth group, the drug was delivered in the middle phase of the dark phase, and in the last group, the fifth one, the drug was delivered at the end of the dark phase. So we can see that the percent mortality was highest when drug was administered at middle of the dark phase, when the mice were most active, and the percent mortality was the highest. However, if you look at this picture, the percent mortality was minimum when it was delivered during their rest span or in the morning hours. It seems that the drug was better tolerated in the morning hours or in the rest span, whereas it is least tolerated by the animal during the middle phase of the dark. So then they replicate the same experiment in another month, and the protocol was the same, dose was the same for all the group, and in individual group there were 15 mice were kept, and then we can see that the result or the outcome of the second experiment was the same, that the mortality was maximum during the middle of the dark phase. So that clearly indicates that the same drug has different effectiveness at different time point of the day. So this experiment give rise to the concept of the chronotherapy. So here we can define the term chronotherapy, that it is a clinical strategy that harmonizes medical treatment with the patient's biological rhythm to have the maximum desired and the least undesired effect. So there is a temporal dissociation in cell division. The cell division in the healthy cells is different from that of the cell division of the cancer tissues, and this provides the main rationale of the cancer chronotherapy that aims at minimizing the treatment toxicity, that is the undesired effect, while maximizing efficacy, that is the maximum desired effect, through proper timing treatment delivery. So the idea of the chronotherapy is that the organisms, including the human beings, they respond differently to same treatment dose for a specific disease when administered at different time points such as day or month or year, as we have seen the example of the Saving et al. In that at different time point of the day the treatment dose have the different effect, and then organisms including the human being, they exhibit rhythm of varying frequencies in susceptibility or the sensitivity to a drug or any kind of treatment. It means the sensitivity to a drug in the morning may be different, in the afternoon it may be different, in the evening hours it may be different. So this is the idea behind the chronotherapy. So if treatment is given using the only single drug, so then it will be called as the chronomonotherapy. But if multiple drugs are given for the treatment, so then it will be known as the chronopolytherapy or the group chronotherapy. And if radiation is given in relation to time, that it will be known as the chronoradiotherapy. Now this concept of the chronotherapy is widely used in the clinics for the treatment of several ill diseases such as the cancer, heart diseases, asthma, arthritis, allergies, sleep disorders such as the delayed and advanced sleep phase syndrome, that is known as the DSPS and ASPS, and then the seasonal affective disorder, that is also known as the mood disorder. So in my lecture I will be focusing main on the cancer chronotherapy. So then, as in previous slide I told that there is a harmonization between the patient's clock with the medical treatment. So then how can we study the biological clock? So we cannot study directly the SCN, suprachiasmatic nucleus. For that we need to study some of the important biological functions that work as the hands of the biological clock, and these are known as the best marker rhythm. Number of marker rhythms such as the core body temperature, melatonin, cortisol, and the rest activity rhythm, they reflect the functioning of the biological clock. So among all these, rest activity rhythm has been widely used particularly for chronotherapy, for the study of the chronotherapy. So this rest activity has been they shown as they are endogenous in nature. This is considered as the major circadian clock output. We know that the sleep wakefulness or the rest activity is the most important physiological function, and another important feature of this rest activity is that multiple sampling, that is the day and night, throughout the day and night we can have the multiple samplings of the rest and activity. Another important feature that it is the non-invasive technique. So how can we monitor the rest activity? So we have the number of devices, that is known as the Actigraphy devices. These are some of the examples: Activas 64, Activas Spectrum, Acticall, Activas 2, Basic Motion Logger. So these are the devices that we wear in the wrist of the non-dominant hand for monitoring of the rest activity. So these actigraphy devices consist of an accelerometer. Thus this accelerometer, it is a piezoelectric set that is capable of converting movements to measurable electrical signals, that is movement in the wrist. So this wrist movement, it converts into the electrical signals, and for recording of or for storage of these signals, these actigraphy devices also consist of a programmable memory chip for storage of this data, that is the electrical signal. Now these are the output of the active watches. Here you can see that this output is known as the actogram. So this is illustrative example of one human individual. So as we are diurnal in nature, so we have lots of activity during the daytime and there is no activity during the night time. So we can say that our rest activity rhythm is very robust, that during the daytime we have lots of activity and during the night time there is no activity, almost zero to no activity. That indicates that our rest activity is very robust in nature and that reflects the proper functioning of our biological clock. And these are the other, these are again the actograms that is derived from the active call or active watch spectrum or active watch AW2. So these are the universal output. Using this active watch devices, we can always have our rest activity profile. So then, simply having this rest activity profile, definitely it will give the pattern of our rest activity. But if there is some disturbances in this rest activity, so only visualization of this actogram is not enough. We need to validate this rest activity profile using some statistical technique, and for this, using this statistical technique, we can derive or we can determine some of the rhythm parameters such as the measure, amplitude, acrophase, mean activity, autocorrelation coefficient, dichotomy index, and by using all this rhythm parameter, we can know whether rest activity rhythm is robust, persist, or there is some alteration. So I would like to explain some of this rhythm parameter. Measure is nothing but a rhythm adjusted mean value. Amplitude, this is the half of the total predictable change in a rhythm. This is the maximum, this is the minimum. So this is the double amplitude, and if we have this double amplitude, so it will be known as the amplitude. That is the half of the total predictable change. Acrophase is the timing of the highest value with reference to midnight. This is the biological function, and at which particular time the biological function is maximum, that reflects its acrophase. That is the time. And period is the duration of the one complete rhythmic cycle. So how much time it takes to complete one cycle, that is known as the period. And mostly in case of if everything is perfectly okay, so then period will be 24 hours. So these are the 4 important rhythm parameters. In case of the rest activity rhythm, we always use 2 important parameters, that is the autocorrelation coefficient and the dichotomy index. So this autocorrelation coefficient, that is nothing but a measure of the regularity of the activity pattern over 24 hours from one day to the next. Here we can see that going to bed time, it is very particular in day by day. Similarly, the wake up time is very particular from day by day. So it shows that there is a consistency in the rest activity pattern. So that indicates that rhythm is very perfect, and again it reflects the proper functioning of the biological clock. And these values of the autocorrelation coefficient varies between -1 and +1, and value if it is near to +1, that clearly indicates there is a very good rhythm in the rest activity. The important variable is the dichotomy index, that is the distribution of the activity during the rest and wakefulness span. And this dichotomy index is a robust, well characterized and clinically meaningful parameter to estimate individual circadian rest activity pattern. Nowadays, this dichotomy index has been it is used for prediction of the survival of the cancer patient. So we can imagine that how much it is useful to determine the dichotomy index. So we can simply compute it as the percentage of time of in-bed activity when the subject is in bed. So we compute the activity during the in-bed time that is below the median level of activity when out of bed. When the subject is out of bed, we compute the median of the activity, and then we compare how many counts during the in-bed is lower than this median of the activity. So thus it essentially assesses the reduction in physical activity while in bed compared to then when out of bed. If there is number of activities which are greater than the median value, that means it reflects the disruption in the rest activity rhythm, that there are number of activities during the night time, and that reflects there is a disruption in the sleep wakefulness cycle, and that again reflects the improper functioning of the biological clock. So overall, dichotomy index provides us the information about sleep quality, and it ranges from 0 to 100%. In normal dichotomy index that approaches 100% indicates restful sleep and regular daily activity. In case of healthy individuals, it rarely goes below the 98%. This is one example of computation of the dichotomy index. Here, as we separate the activity when the subject is in bed and when subject is out of bed. So we compute the median when the subject is out of bed, and then we count how many in bed activity are below this median level. So we can see this is one hypothetical example. So we can see that out of 15, 14 activity are below the 23, and the ratio of this two is 93.33. That indicates that it is a good sleep wake cycle, or the sleep quality is good. If there is a reduction in the IO, so that it will indicate that there is some problem with their rest activity. So we know that in case of cancer patient, there is a disruption of rest activity rhythm, and it has been documented in patients suffering from breast cancer or metastatic colorectal cancer or advanced non-small cell lung cancer and head and neck cancer. If there is disruption in the rest activity, so this abnormal or disrupted circadian rest activity rhythm is associated with the higher progression of the cancer. The cancer progression will be more if there is a disruption in the rest activity rhythm. The survival rate will be lower if there is a disruption in the rest activity rhythm, as well as the quality of life of the cancer patient will be poor if there is a disruption in the abnormal circadian rhythm. So then these are some of the examples of the rest activity rhythm disruption in cancer patients that we carried out in our lab. So there is comparison between the control subject and the cancer in patients for the rest activity rhythm in both male and female. So in case of the control subject, in male control subject, we can see there is a clear-cut distinction between the rest and activity. During the night time or during the sleep period there is no activity, but during the daytime there is lots of activity. There is a clear-cut distinction. But in case of the cancer patient, age-based cancer patient, we can see that there is a complete disruption of the rest activity rhythm. We cannot identify, looking if we hide this time window, so we cannot differentiate between the day and night, because there is continuous activity during whole 24-hour time scale. Similarly, in case of female, again in control subject there is a clear-cut difference between the day and night. During the night time no activity, during the daytime lots of activity. But again in female cancer patients, this day and night difference between the activity is abolished. There is no clear-cut pattern of the rest activity. This clearly indicates that there is a disruption of the rest activity rhythm in cancer patients. This is the example of inpatient, that is the breast cancer inpatient, across the chemotherapy cycles. During the first chemotherapy cycle, the rhythm was maintained. However, as chemotherapy cycle progresses, so during the chemotherapy cycle 6, we can see that there is a activity level is minimized, as well as there is no distinction between the day and night. So that clearly indicates with the progression of chemotherapy cycle there is a disruption in the rest activity rhythm. In case of outpatient, here again we can see that during the chemotherapy cycle 1 rhythm is maintained, but during the chemotherapy cycle 6 or with the progression of the chemotherapy cycle there is a disruption in the rest activity rhythm. It means the cancer disease as well as the chemotherapy, both they aggravate the cancer progression itself. So this is another study that we performed in Hospital Paul Bruce along with the Dr. Francis Levy. In this particular group, we included 436 metastatic colorectal cancer patients, and then here we can see that there is inter-individual variations in the rest activity profile of the cancer patient. In one patient, rhythm was maintained and dichotomy index was 100%. However, in another patient, the dichotomy index was reduced, that is up to 77.8%, and we can see that this rest activity rhythm it is completely abolished. So we find inter-individual variations in the cancer patients. Then we considered dichotomy index for prediction of the overall survival in these cancer patients, and we divided the cancer patients into two groups on the basis of the dichotomy index. We take the median of the dichotomy index as the reference point, and on the basis of this we divided the patient into two groups. In one group, those patients who have the better rhythm, that is the high dichotomy index, it means they have the robust rhythm, and the another group where dichotomy index was below this median level, and we considered them as the patients with the disrupted rhythm. And then we find out that the median overall survival was 21.6 months in the group who have the robust rhythm, whereas in the patients who have the disrupted rhythm, the median overall survival was 11.9 months only. Then we also computed progression-free survival, again taking the median dichotomy index, and then we find that median progression-free survival was 9.3 months in the group who have the robust rhythm, that is the higher dichotomy index, whereas it was reduced to 5.8 months in the group who have the disrupted rhythm. So that clearly indicates that dichotomy index is a better predictor of the that is the survival. So and this is the representation of the rest activity rhythm. So we can see that how this rest activity rhythm could be important for the prediction of the functioning of the biological clock. So I will be speaking about the chronotherapy of the cancer chronotherapy with reference to rest activity rhythm in my next lecture.